Welcome guys, welcome to my channel Adhyan with Flow. Okay, in our previous video we already done a greatest important thing, amino acid, in a very huge form. Okay, you have to watch them at least one time because much beneficial according to your competitive exam, much beneficial. Okay, now again this is your headache if you not to study this. Okay, in previous video we skipped this mechanism. Okay, and what is zwitter ionic? I'm not taking your much time because we want to start lipid. In previous video during amino acid we forget this reaction and we explain all the thing. Okay, so this is the amino acid structure, ncc rules. Okay, ncc rule. For detailed explanation you have to watch my amino acid, and whoever watch this my video they can better understand what is ncc rule. Okay, now carboxylic group have tendency to release their what electron, their hydrogen, so they become negative in charge. But this amino acid, this is work as a GF. It does not give any kind of paisa. This is the BF. They give paisa. They give money. They expend money. If we provide some acidic medium, they donate their hydrogen. But she still not give their hydrogen. But in critical condition, if we put amino acid in basic medium, so then because he release their all money, BF give their all money. Now this time to pay. Now this time to turn for GF, and they donate their hydrogen. When now, and someone asks what is zwitterion, then what you say? Then what you say that time? Zwitterion, when the overall charge of the amino acid become 0. Here this is the amino acid, but here the overall charge on this amino acid is positive. So this is not a zwitterion. Here overall charge on this amino acid is negative. Look at here, there is no charge, but here is a charge. So this is the negative. But here look at, there is a one positive charge and one negative charge. Positive, negative, they are cancellate each other and they have 0 charge. 1 plus charge, 1 minus charge. So they cancel out and they become 0. So this form is deuterion form. I escaped to teach you this thing. Now your amino acid is complete. Now let's move towards the lipid. I know guys, lipids are macromolecule. Still, they are macromolecule and they cross plasma membrane, guys. Why? Because they are hydrophobic. Lipid are hydrophobic. And PM, plasma membrane, are also hydrophobic. So they can easily cross plasma membrane. Why? Because plasma membrane made up with phospholipid, guys. So they how can stop the lipid? How can stop the lipid? Because they made up with lipid, brother. So they can easily cross. So they can easily cross plasma membrane, cross EM. Okay, all right. Now let's move further. They also contain R group, guys. It may be methyl, ethyl, and huge number of carbon chain 1 to 19. Okay, I'm telling you further, guys. Relax. Lipid have 3 branch. Phospholipid, I'm taking little bit. Phospholipid, glycolipid, and sterol. They are lipid form. They both of them, they can cross plasma membrane. They made up with 4 components. Like they made up with alcohol. This is not important. Fatty acid, phosphate group, and some R group. I told you na, R group, it may be 1 carbon, it may be 2 carbon, it may be 3, 1, 4, 5, 6, 7, 8, 9, 10, 1 to 19. Glycol, look at now, phospholipid means they have phosphate group. Okay, they have phosphate group. They have lipid group, they have lipid group, and they have R. Now, what is glycolipid? Glycolipid means carbohydrate plus lipid. Carbohydrate plus lipid means they have fatty acid, they have alcohol, and they have what? Sugar group. Carbohydrate, na? They have sugar group. Clear, guys? They also contain 1 sugar. They also contain more than 1. If someone asks what is the example of glycolipid, they contain 1 sugar. You can tell them cerebroside. And someone asks the name of the glycolipid who contain many sugar, then you have to say ganglioside. This is not in your NCERT. I'm taking you beyond the level of NCERT. According to competitive exam, this thing is important. Compare than this thing. Okay. Now steroid, they are ring-like structure. That's it. They are ring-like structure. They contain rings. They contain rings. Cholesterol, remember cholesterol, cholesterol. They are lipid. Okay, now here is writing some that some have 16 carbons, some have 20 carbons. I'm giving you detailed example of 14, 15, 16, 17, 18 carbon examples. Okay, I'm writing here some example of 14, 15, 16 carbon. According to your book, book give arachidonic acid, they contain 50 carbon. At R group, they contain 50 carbon. Some example of palmitic acid, they contain 16 carbon. I'm giving you more example. Look at me here. I'm writing here palmitolic acid, they contain 60 carbon. oleic acid, they contain 18 carbon. Example, I'm giving you example. Myristoylic, they contain 14 acid guys, acid. Linoleic acid, they contain 18 carbon. Now arachidonic, they contain 20 carbon. So these are example of carbon. Guys, these are the unsaturated, okay, fatty acid. Unsaturated means they have 16 carbon. Yes, I know they have 16 carbon, 18 carbon, 14, but they also have double bond. There is chain, like there is a chain, and here somewhere double bond is present. So if they contain double bond, we will take it as a unsaturated. This is already here guys, that's why I'm teaching you what is unsaturated. One or more carbon double bond. Okay, now I'm giving you some saturated fatty acid, means lipid examples. Guys, remember my thing, these are important for your examination. Don't take a easy, okay? Please don't take a easy, these are important. Topper will understand this thing, that they are important according to competitive exam. This is not your 11th biology class exam. This is now this term to your competitive exam. You want to fight, you have to fight competitive exams. Now some example, they does not contain any kind of bond, single bond. Look at here guys, we are saturated without double bond, they contain only single bond. There are some example, I'm telling you one by one. Butyric acid, they contain how many carbon? They contain 4 carbon, but they does not contain any kind of double bond. Next is caprioic, 6 carbon, they does not contain double bond. Next is caprylic, 8 carbon, okay. Copric, they contain 10. Wait. Lauric, they contain 12. Or myristic, they contain 14. Carbon. Now palmitic, they contain 16 carbon. Look at here guys, 16 carbon, palmitic, 16. I'm not saying palmitolic, this is palmitic. This is your example in your NCERT, okay. Now stearic, 14 carbon. Now arachidinic, they also have 20 carbon. If I say arachidonic, then they have 20 carbon, but they still contain double bond. Aridinic, they are 20 carbon. Yes guys, they are 20 carbon, but they don't contain double bond. Okay, in palmitic they contain 1 double bond, and oleic they contain 1, and they contain 2, and arachidonic they contain 4 double bond in their chain. In their 20 carbon chain, they contain 4 double bond. In their 20 acid long chain, they contain 4 double bond. They contain 1 double bond. They contain 1, 2, and they does not contain any kind of bond. They are single bond structure and they contain double bond. Now you are clear what is unsaturated and what is saturated. Now what is the difference, like if we talk saturated, unsaturated, I'm writing here. What is the special feature of saturated fatty acid? This is the part of amino acid guys, fatty acid. Look at here now. Look at here, fatty acid, lipid part. Now unsaturated. First difference is single bond, they contain single bond and they contain double bond. This term is clear? Okay. So they contain single bond. So they arrange in this manner. No gap. So they become more rigid. No gap. If I draw phospholipid bilayer this time, then they are made up with saturated acid. They are more tightly compact. So not any kind of molecule cross them. Means fluidity less. Means no one can cross this plasma membrane. If fluidity is less, means their melting point is high. Not easy to break this thing. Okay, now let's talk about unsaturated acid. Double bond means they have double bond. So somewhere bond is strong and somewhere bond is weak. So here this is the structure. They produce some kink because they have double bond. They produce kink because they have double bond. So their bonding is less. Strength is less. So their fluidity, anyone can pass. Fluidity is high. Means anyone can pass them. Say they are loosely arranged. Because kink is present, because double bond is present. So melting point is very less. If I give little bit temperature, they can break. But here this bonding is very tight. I'm giving less temperature, they don't break. If I give high temperature, then they break because fluidity is very less. And here fluidity is high. Anyone can cross them. So this is guys important according to your NCERT. Whatever I told you now, everything is written here. Okay guys. Okay now. Where is okay? This is the carbohydrate sugar group. Sugar group plus lipid. We can call it as a phospholipid. No. We call it as a glycolipid. Phospholipid means they contain phosphate group. This is the palmitic structure, 16 carbon. Glycerol, fatty acid. These are chain of fatty acid. Cholesterol, guys, example of sterol. Phospholipid. Look at that, guys. This is everything in your book, guys. You have to remember. I make a sequence for you. I'm arranging the order. They are mesh, but I'm arranging order for you. Monoglyceride means one chain is present. Monoglyceride means one chain. Diglyceride, two chain. Two chain. Triglyceride means three chain. One, two, three. This is the triglyceride. If I say diglyceride means here only two chain. If I say monosaccharide, it means one chain. That's it. Can I repeat again, guys? Can I repeat again? If I take one chain, we call it as a monosaccharide. If we take two chain, we call it as a disaccharide. If we take three chain, we call it as a trisaccharide. This is the arrangement of triglyceride. So guys, that's all about lipid. And these are nucleic acid. So nucleic acid, I'm gonna teach you nucleic acid in our next video. Okay, I'm not going so fast. Okay, relax guys, don't be panic. So today I complete your lipid. In our previous, I complete your amino acid. In next video, we will complete our nucleic acid. Now, guys, here a single word is important. My single word is also important. This is huge. No one can teach you this. No, in your classroom, no one can teach you this. This is the simplest form to represent what is the saturated, what is the unsaturated. You have to revise, guys, how fluidity is decreased, and if fluidity is decreased means melting point high, what kind of phenomena present. These phenomena present in saturated fatty acid because they arrange in tightly manner. So guys, thank you. Thanks a lot for listening me. And what are you waiting for, guys? Subscribe my channel. Okay, if you want to clear your concept, if you want to explore your knowledge, subscribe now, guys. Thank you. Thank you for listening me. Okay, we will meet in our next video with nucleic acid. Thank you.